Students, continuing with our lecture series on medicinal chemistry. This is our 23rd lecture on medicinal chemistry, and from this lecture we will start with antibacterial drugs. And in this series of antibacterial drugs, in the first lecture we will discuss about inhibitors of cell wall synthesis. Now, antibacterial drugs basically is a general term which represents a group of drugs which includes antibiotics, antifungals, antivirals, and antiprotozoals. Out of all these category of drugs, antivirals we have already discussed in few lectures, and other antibacterial drugs we will discuss one by one. And first, under the category of antibacterials is mainly we will discuss about penicillins, phossporins, and other naturally occurring antibiotics. The term antibacterials, as you have seen, it includes antibiotics also, but generally we refer antibiotics as the substances which are produced by microorganisms. And as we all know that the story of antimicrobials began with the observation of Sir Louis Pasteur and Julius Francis Chopra, who discovered that a type of bacteria could prevent the growth of another type of bacteria. So, the important among these antimicrobials is the discovery by Pasteur, that is penicillins. Now, basically, the antimicrobial drugs have an important quality, that is selective toxicity, which means that these types of substances, that is antimicrobial drugs, they will selectively kill the growth of microbial targets, or it will inhibit the growth of microbial targets, whereas it will not affect, either will not affect, or it will have a minimum effect on the cell of the host body. And this selective toxicity is achieved because of the difference between metabolism and structure of the microorganism and that of the human cell. For example, penicillins and cephalosporins, they have been found to be the most effective antibacterial or antimicrobial agents, and what they do is they prevent the synthesis of peptidoglycan, thereby inhibiting the growth of the bacterial but not the human cells. There are four major sites in the bacterial cell which are sufficiently different from the human cell, and these four sites, they are cell wall of the bacteria, cell membrane of the bacteria, ribosomes of bacteria, or nucleic acid of bacteria. All these they have uh, structural differences from that of a human cell. So because of that, these four sites, they are used by the antimicrobial drugs to inhibit the growth of bacteria. Now, as far as mechanism of action of these antibacterial drugs, as I have told you, the mode of action or mechanism of action of these drugs is the way in which a drug affects the microbes at the cellular level. So if the microbes, they are affected at the plasma membrane level or at the cell wall level, or RNA synthesis, DNA synthesis, or ribosomal synthesis, or the metabolic pathways of the bacteria, they are affected by these antimicrobial drugs. The drugs which affect the plasma membrane of the bacteria, they are polymyxins, daptomycins, Glycopeptides, and colistin. Whereas those bacterial drugs which affect the cell wall of the bacteria, they are penicillins, Phossporins, monobactams, beta-lactams, Carbapenams, Vancomycins, Glycopeptides, and bactracin. The drugs which affects the RNA synthesis in case of bacteria, they are rapamycins or rifampin, whereas sulfonamides, fluoroquinolones, ciprofloxacin, levofloxacin, and moxifloxacin, they affect the DNA synthesis in case of bacteria. The activity of ribosomes is affected in bacteria, wherein these drugs, they inhibit the protein synthesis of the ribosomal subunits. Either they will affect the 30th subunit or 50th subunit in case of bacteria. Tetracycline and aminoglycosides, they affect the small unit, that is 30th subunit of ribosomes, whereas macrolides, lincosamides, and chloramphenicol and oxazolidines, they will affect the 50th subunit of ribosomes in their mode of action. So we will discuss these one by one. First, in this lecture, we will describe mainly the inhibitors of cell wall synthesis. So, as I have already told you that these types of antibacterial drugs, they will affect the synthesis of the cell wall in case of bacteria. What they will do is they will block steps in the biosynthesis of peptidoglycan. Thereby, these cells, they will be made susceptible to osmotic lysis. Therefore, bacteria will be killed by these types of steps. Uh, these human cells, they don't make peptidoglycan, so therefore this mode of action is an excellent example of selective toxicity. So peptidoglycan will be affected only in case of bacteria, and in the human cell there is no peptidoglycan, so this will not be affected. Most important among these inhibitors of cell wall synthesis, they are beta lactams, and the most common example of beta lactams is penicillin, which was the first antibiotic discovered by Louis Pasteur. These beta lactams, they not only include penicillins but cephalosporins. They will also have these types of beta lactam rings. So penicillins, they will have this type of beta lactam rings. Cephalosporins, they will also have these types of rings. Then monobactams, they will also having beta lactam ring, and then carbapenem type of antibacterial drugs, they will also have beta lactam rings. And this beta lactam ring, this forms the central structure in all these types of drugs. And these drugs, they are differentiated due to the presence of this R group, and if this R group is this benzyl, then this is penicillin will be called as penicillin G. If this group, then penicillin B. If R is this group, ampicillin. R is this group, these are same. Ampicillin and amoxicillin, only difference is at that fourth position we are having a hydroxyl group, and if this substitution is there, then this is called as methicillin. So all these penicillins, they have almost similar structure, and only point of difference is the substitution at this arc. Now, what these beta-lactam antibacterials do, they block the cross-linking of peptide chains during the biosynthesis of new peptidoglycan in the bacterial cell. So, therefore, by blocking this process, beta-lactam structure is similar to the structure of peptidoglycan subunit component. So, therefore, it is recognized by cross-linking transpeptidase enzyme, which is also known as penicillin binding protein. And because of this, the beta-lactam ring must remain unchanged for these drugs to retain their antibacterial activity. As I've told you, this is the basic structure of penicillins, and here in case of Sphalosporin, this is a six-membered ring instead of five-membered ring. And all these penicillins and sphalosporins, they will act by inhibiting transpeptidase enzyme, and this transpeptidase enzyme catalyze the final cross-linking step in the synthesis of peptidoglycan, that is bacterial cell wall. So, in addition to blocking this synthesis of peptidoglycan, there are two additional factors which are involved in action of penicillin. The first factor is that penicillin binds a variety of proteins in the bacterial cell membrane and cell wall, and these proteins, they are called as penicillin binding proteins. And these are transpeptidase. The other function in the synthesis of peptidoglycan, that is what takes place. And these changes in penicillin binding proteins, they are responsible for an organisms becoming resistant to penicillin. Then second factor is that itolytic enzymes, which are called as murine hydrolases. This is murine is again a synonym for peptidoglycan. They are activated in penicillin treated cell, and they will degrade the peptidoglycan. Whereas some bacteria, that is SRAs, they are tolerant to action of penicillin because these itolytic enzymes, they are not activated. Then in those cases, when cells are growing, new peptidoglycan is being synthesized, the transpeptidation occurs, and however, in case of non-growing cells, no new cross-linkages are required and penicillin becomes inactive. So these are the activity of different types of penicillin. For example, penicillin G and penicillin V. They are effective against gram-positive cocci, gram-positive rods, Neisseria, Treponema, then many other anaerobic bacteria, then gram-negative rods. They are also affected by these types of penicillins. Then ampicillin or amoxicillin, they also affect certain gram-negative rods such as Haemophilus influenzae, Escherichia coli, Proteus, Salmonella, Shigella, but they don't affect Pseudomonas aeruginosa or Klebsiella pneumoniae. Similarly, ticarcillin, this affects Pseudomonas aeruginosa, whereas piperacillin, this is having activity similar to ticracin, but with greater activity against Pseudomonas aeruginosa and K. pneumoniae. Then naphthalene or dicloxanilin, they affect the Staphylococcus aureus, whereas methicillin, this is narrow spectrum antibacterial which is active against gram positive bacteria only, and other strains which produce penicillinase. Similarly, cephalosporins, as I've told you, only difference between cephalosporin and penicillin is instead of 5-membered rings, cephalosporins they are having 6-membered, and they are synthesized semi-synthetic antibacterial cephalosporins. They are synthesized from 7 amino cephalosporinic acid, whereas the penicillins, they semi-synthetic penicillins, they are synthesized from 6 APA, 6 amino penicillinic acid. These are some of the semi-synthetic cephalosporins which are active against different types of bacteria, as these have been detailed out. Then some cephalosporins, they become inactive because of beta-lactamases with this enzyme, which is an important clinical problem. So in such cases where these cephalosporins they are inhibited by beta-lactamase activity, then a combination of cephalosporins they will prevent the inactivation of the cephalosporin. For example, tazobactam and avibactam. This is avibactam and this is tazobactam. When they are given in combination, they will be active against beta lactamase activity. Then uh, Spetazoline and tazobactam, they are also given in combination for treatment of intra-abdominal infections and complicated urinary tract infections, and they will also prevent inactivation of beta lactamase activity. Whereas this spetazidine and avibactam, they are also given in combination to prevent the beta-lactam inhibitor activity. Then next category is carbapenems. As I've told you, carbapenems are again beta-lactam drugs which are structurally different from penicillins and cephalosporins. And most commonly used is carbapenem is imipenem, having this structure. So this is having a beta lactam ring with a 5-membered ring where the sulfur is missing in comparison to penicillin. So this imipenem is one of the widest spectrum of activity in case of beta lactam drugs, and excellent bactericidal activity against many gram positive, gram negative, and anaerobic bacteria. It is also effective against most gram positive cocci, gram negative cocci, gram negative rods, and various anaerobics. Similar to imipenem, these are other types of carbapenems which are effective against different types of bacteria. For example, ertapenem, the structure is similar to imipenem with the substitution at this place, but this is not effective against Pseudomonas aeruginosa. Similarly, doripenem or meropenem, they are also similar to imipenem but slightly more active against gram-negative rods in comparison to imipenem. Then is monobactams. Monobactam, as I have told you, they are also beta-lactam drugs. They are monocyclic in nature. No other 5-membered or 6-membered ring is attached to the beta lactam. So without an adjacent sulfur containing ring structure, and most common is astrionum. This is the monobacterium which is most commonly used, and it has shown excellent activity against many gram-negative rods, gram-positive rods, and anaerobic bacteria. And advantage of this monobacteriums is they are resistant to most beta lactamases. So therefore these are very useful in patients who are hypersensitive to penicillin because there is no cross reactivity. And then there are some naturally occurring antibacterial agents, glycopeptides. Most common example is vancomycin. Vancomycin is a glycopeptide that inhibits cell wall peptidoglycan synthesis by blocking transpeptidation, but by a mechanism different from that of beta-lactam. So what vancomycin do is it binds directly to D-alanine portion of the pentapeptide, which blocks the transpeptidase from binding. Whereas, we have seen that beta-lactam drugs, they bind to inhibit the activity of transpeptidase itself. Whereas they will inhibit the activity of portion of the pentapeptide. So they are effective against gram-positive bacteria and most useful in the treatment of infections caused by SRS that are resistant to penicillinase resistant penicillins, nafcillin and methicillin. Because methicillin resistant SRS they are effective against. Then this is also effective against treatment of infections caused by S. epidermis or S. pneumoniae. Then enterococci with partial or complete resistance to vancomycin have been recovered from the patients. Then some derivatives of vancomycin, they have been also synthesized. For example, telvacin. Uh, telvacin is this one. This is again a derivative of vancomycin, but their mechanism is same. They will inhibit the peptidoglycan synthesis and they will disturb the bacterial cell wall membranes. And this is useful for treatment of skin and soft tissue infections, especially those caused by uh, MRSA, methicillin resistant SRS. Then oritavancin or oritavancin and dalbavancin. They are again glycopeptide derivatives of vancomycin and teicoplanin respectively, and these drugs they will again inhibit the transpeptidase and transglycosylases which are required for the synthesis of peptidoglycan of the gram-positive bacteria. So they are effective in the treatment of SRS. Then is bactracin. Bactracin consists of a group of structurally similar peptide antibiotics. They have been isolated from Bacillus subtilis. They block the activity of a specific cell membrane molecule that is responsible for the movement of peptidoglycan precursors from cytoplasm to the exterior of the cell, and therefore they will prevent the incorporation into the cell wall. Then these bactracins, they are effective against a wide range of bacteria, including gram-positive organisms found on skin such as Staphylococcus or Streptococcus. So these are the references which have been used in preparing this lecture. Thank you very much.